The next speaker, Dr. Andreas Vlachos. Andreas, are you with us?
Thank you. It's great to be part of this and share some thoughts and also some of the ongoing work in my lab in Freiburg. So another one? Yes. So during the last couple of years, we've been collaborating with Alexander Oppitz and also with the groups of Kilian Kweiser and Peter Jedrzejka to develop computational tools that will enable us to not only predict and standardize the electric fields, but ideally, of course, also some of the desired biological effects across scales and also across models and individuals. So the way that we are currently trying to inform these models, so about the biological effects that certain electric fields are inducing, is we use dedicated microfluidic chambers that give us full control about the electric fields that are induced in our target tissue. These are usually acute cortical slices or cultured organotypic tissue cultures. And as you can see down here, these chambers are transparent, so we can use microscopy to visualize functional and structural changes as we turn on and off the electric fields. And we can also use optogenetic approaches to actually modulate and modify the state of these preparations. Another interesting feature of that model is that, and this is shown on the right-hand side, that we've implemented actually a magnet, and so the cultures, the slices are placed on stainless steel, so we can rotate also the target tissue within the electric field and actually also look at different orientations. So we started looking at direct current stimulation and alternating current stimulation using these approaches, and also TMS-like stimulations. So next slide. And the result that we obtain from these preparations, we compare with experiments at the setup that you see on the left-hand side. So this is a conventional figure-of-8, 70 millimeter inner diameter coil that is placed below a microscope. We took great care to design the setup in a way that pulsing the coil will not destroy the setup. So we can actually now look at our tissue again with a microscope and started comparing the results with the conventional TMS coil with what I've just showed you with the microfluidic chambers. Most of our work is done in animals, so in rats and mice. We actually get quite some interesting data on differences between rat cultures and mouse cultures, despite the fact that we standardize the electric fields. So biological effects apparently matter. But one important aspect, after all, at the end is that because we want to stimulate the human brain, we would like to get living human cortical tissue into the setup. So a while ago here in Freiburg, we teamed up with our colleagues in neurosurgery and established a framework that enables us to actually get living human cortical tissue from neurosurgical resections. So this is small pieces of cortex that is otherwise thrown away. We can put those small pieces of cortex into the microfluidic chambers that I showed you at this setup that you see here, and start getting some interesting cross-species results, so to say. Also considering that there is some exciting work coming up, for example, showing from Moritz Helmstedter's lab, showing that there are 2.5 more interneurons in the human cortex, 10 times more interneuron to interneuron connections. So we're trying to actually use this type of information, and there will be more and more coming up in the literature to implement this in our biological models. Finally, next slide. And Maron Bixmo was already mentioned, I think it will be important to implement also non-neuronal cells. So what you see here is actually an experiment now in an intact animal. So we use TMS also in anesthetized mice in this case. We're not so much concerned about focality, but just about the electric fields in a certain brain region. So this is the mouse MPFC. What you see in the upper part are in magenta microglia. In the lower part, we removed the microglia. Just removing the microglia, although nothing changes on the baseline condition, at least from what we can tell at the moment. As you can see here, the biological effects are different, despite the fact that the electric fields are the same. So in one case, with the microglia, we see some potentiation of synaptic transmission. In the other case, in the absence of microglia, we don't see them. So I fully agree with Maron Bixmo that we really need to take also the other cell types in the brain into consideration when it comes to dose response curves. Thank you.
Thank you very much, Dr. Vlachos.